papers on Mycobacteria. He wants to invite him to see his presentation here. I'm very happy that we make a video with this, and Michel can benefit for this as well. But I'm very happy and very proud that we're here to talk about something that we're still facing, and we have a team working speaking. So very welcome, and we're happy to hear from you.
Thank you very much for your kind introduction. It's a pleasure for me to be here. I enjoyed my stay in Marseille at the Old Harbor. This morning I had a nice walk around at the fishing market, and it was colorful and pleasant. However, I also am pleased to be here because I consider the subject I'm going to present as very important. And also think this subject is underestimated. Not many people are working on the clinical relevance of non-tuberculous mycobacteria. People are familiar with tuberculosis, but not with the disease that mimics tuberculosis, infection by non-tuberculous mycobacteria. I'm the head of the National Tuberculosis Reference Laboratory in the Netherlands at the RIVM in Bilthoven. And I also have close collaboration with the Radboud University, where I am a professor. And I especially work with the lung center that treats patients with multi-drug and extensively drug-resistant tuberculosis, as well as patients with complicated infections with non-tuberculous mycobacteria. The work I'm going to present is also in close collaboration with other laboratories and hospitals in the Netherlands. Well, there is one other person I should mention in relation to the work on NTMs, and that is Jacco van Inge. Jacco van Inge in the last years published more than 30 scientific publications together with us. The average impact factor was nearly 5, and he obtained his PhD degree cum laude at the Radboud University in September 2009. If you have a special interest in this subject, please let me know. Then I can send you a copy of his thesis. Tuberculosis is still a considerable problem in the Netherlands as it is in the rest of Europe. In the Netherlands we have about 1,000 new cases every year. The relative contribution of TB cases in first-generation immigrants is increasing. It's now about 73% of the total number of cases. While the number of TB cases in the natives is decreasing, it's about 19% at the moment. Until 2006, we received all isolates of non-tuberculous mycobacteria at the RIVM. Since then, we asked payment for that kind of service, and the number is decreasing, unfortunately. But you see something interesting. While the number of isolates of tuberculosis complex isolates is decreasing in the period of 2002 to 2006, the number of isolates of NTM is increasing. And that is something that we have noticed in other Western countries too. Actually, the genus Mycobacterium can be divided in two groups as shown over here. We have the Mycobacterium tuberculosis complex that comprises bacteria that are genetically highly conserved. Actually, if you look at the subgroupings within the TB complex, well, you can even not speak of separate species. Actually, they are only subspecies of the same grouping of bacteria, with one exception, and that's Mycobacterium carnetii. We described Mycobacterium carnetii in 1987, and actually Mycobacterium carnetii has the highest degree of similarity with the hypothetical ancestor of the whole TB complex. And the rate of divergence among strains of Mycobacterium carnetii is very large. It's the oldest divergent of the complex. That story is not true for the group of non-tuberculous mycobacteria. If you look at the species of non-tuberculous mycobacteria, there's a high degree of divergence, evolutionary development. But also if you look within species, there is a high degree of diversity. So if we speak, for instance, of Mycobacterium kansazai, we actually group bacteria with quite a degree of evolutionary divergence. I will come back to that. We have to realize that non-tuberculous mycobacteria are all around us. They're present in tap water, surface water, in soil, in milk, products, etc. I remember having a discussion previously with an appreciated colleague, Françoise Portals from Antwerp. She said to me, well, the NAVians that you will find in soil are not the same NAVians that cause severe infections in HIV positives. However, on basis of extended comparisons by the new molecular tools that we have to conclude that the same type of bacteria that cause infections in humans are found in soil and also in surface water, etc. This is an example of Mycobacterium szulgal. Mycobacterium szulgal can cause very severe infections, lung infections as well as skin infections. And you can simply find these bacteria in tap water. And perhaps you think, well, that's an old story. And nowadays, with our high quality tap water, you cannot find any Mycobacteria anymore. Unfortunately, that is not true. We did another isolation of Mycobacteria in 2009 in a place, Nieuwegein, in the central part of our small country. To our surprise, we found a huge variety of different Mycobacteria. Interestingly, we saw so much variety that some of the Mycobacteria we could not recognize on the basis of the molecular tools that we nowadays use. It seems to be there is a distinction between the Mycobacteria present in tap water and the ones infecting humans. However, also the ones that do infect humans were found in tap water. Some words on the disclosure and description of Mycobacteria. In 1882, Robert Koch disclosed Mycobacterium tuberculosis as the causative agent of tuberculosis. I don't know whether you've ever been to Berlin, but the theater where he showed that Mycobacterium tuberculosis, the causative agent of tuberculosis, is still there. It's circular, and he just placed a microscope in the middle of that theater, and he asked the audience to step down to have a look in the microscope to see the causative agent of tuberculosis. Of course, that was a revolution. However, at that time point, people thought, well, tuberculosis is an inherited disease, so they did actually not believe him. But it was, of course, very, very important finding. In the years thereafter, Mycobacteria were found in cows, birds, reptiles, fish, soil, water, etc. And in 1896, Lehmann and Neumann described the genus Mycobacterium. In 1926, Mycobacterium marinum was disclosed that causes fish tank granulomas, as shown over here. We see that type of infections on regular basis in the Netherlands. In 1935, Pinner introduced the term atypical Mycobacteria. In the 1950s, 1950s, Mycobacterium ulcerans was disclosed, causing Buruli ulcer. I have a photograph of that later on. In the 1950s, Mycobacterium avium and Mycobacterium kansazai were described. Mycobacterium avium was associated with severe disseminated infections in HIV positives, and Mycobacterium kansazai is associated with lung disease, especially in minors. From the 1990s on, many new species were disclosed, and actually in the database you can find more than 150 species described at the moment. They're not all valid, because people even describe new species on the basis of one base pair differences, but that is what you can find. Well, Buruli ulcus is caused by Mycobacterium ulcerans, and in particular parts of Africa this is really a huge problem, and even an increasing problem. There are some clues on how Mycobacterium ulcerans is transmitted, but we are not completely sure how that takes place. So it's difficult to prevent the spread of Mycobacterium ulcerans. Well, what is the nomenclature? Well, there have been different namings for a typical Mycobacterium, like anonymous Mycobacteria, environmental bacteria, saprophytic Mycobacteria, opportunistic bacteria. Very fashionable was this naming, Mycobacteria other than tubercle bacilli or moths. Some people still use that terminology. However, since 2008, the official term is non-tuberculous Mycobacteria or NTM. Well, if you look at the database more carefully, at the moment there are actually 130 well-described and accepted Mycobacterium species. And we recently added 5 additional species as shown over here. Mycobacterium novio-magense, riadhense, insubricum, vulneris, and montani. And of course, it is fun to introduce a new species. I will be honest about that. But the only reason why we do that is that there is a difference in clinical relevance between different species. I have an example later on. We, by the way, also introduced a new Mycobacterium tuberculosis complex subgrouping, and that's Mycobacterium aurigis that has been recently published. We have a problem with isolation of NTMs. Actually, NTMs can cause infection, but also contamination, because they arise from environmental sources as stated previously. We believe that transmission from humans to humans does not occur. However, it's difficult to prove that. We have sophisticated typing methods nowadays, so if we, for instance, isolate Mycobacterium malwense from persons of one household, there is an assumption there may be transmission. But if you find identical DNA fingerprints, you are not sure whether there is transmission from one person to the other, or whether they share the same environmental source of infection. So it's very difficult to deduce that. So they're typically opportunistic pathogens. They infect patients with locally impaired immunity, suffering from COPD, for instance, or with a history of tuberculosis. Or they sometimes suffer from systematic impaired immunity, like HIV-positive patients with leukemia or having suppressive medication. So in general, you can say that non-tuberculous Mycobacteria cause a lot of pseudo-infections, but also laboratory and bronchoscope contaminations. So in general, we can say that the clinical relevance of isolation of non-tuberculous Mycobacteria is often unclear. But why do we in the Western world see an increased isolation of non-tuberculous Mycobacteria? Well, undoubtedly, physicians are more aware of non-tuberculous mycobacterial infections. Although, as I speak, there are still laboratories that, if they isolate non-tuberculous Mycobacteria, they just discard them. They even do not have a proper look at the patients, still in 2012. And we have an improved culture method, of course, the liquid culture system. With liquid culture systems, you will have a higher yield of non-tuberculous Mycobacteria. We have an improved identification by our sophisticated molecular tools. And of course, we have changes in the human host. We have the increase in the epidemic of HIV. In general, you can say we have a higher life expectancy, so you have more patients with chronic lung problems like COPD. And there is less cross-protection because there is a decrease in the prevalence of tuberculosis, and there is less vaccination against tuberculosis, for instance, in some Western countries. Some people also claim there may be changes in the pathogenesis of NTMs because of the man-made environments. Perhaps by introducing tap water systems, you are selecting for particular NTMs that you will not find in surface water. They, for instance, like the type of material, and they form biofilms within the tap water systems. And I think this is a very important one. There is an increase in frequency of showering. And showering is, of course, a very effective way of infection if you are susceptible to NTM infection, because you're exposed to aerosols of NTMs, and well, that's almost the most ideal system to infect someone. Well, now some words on the research conducted at the RIVM. Actually, it's the correlation of clinical and laboratory data for a wide variety of different Mycobacterium species. The purpose of that all is to have more insight in the clinical relevance of isolation of NTM. Actually, we had a look at the demographic data of the patients, the clinical data, and the microbiological information. And we used the American Thoracic Society diagnostic criteria, the ATS criteria, to determine whether isolation in these patients was clinically relevant. These ATS criteria are based on symptoms, radiological abnormalities, the bacteriology, so microscopy and culture, and the histology, so the type of inflammation. In principle, these ATS criteria are for M. avium, M. kansazai, and M. abscessus infections. However, you can extrapolate that also to other NTMs. And here you see a part of the criteria of the ATS. They have been published by Griffith, but you can find them on the internet. It's actually regarding the clinic pulmonal abnormalities on the X-ray or CT scan, the exclusion of other symptoms. Regarding the microbiology, well, you need to have 2 sputum samples that yield the same NTM, or you need to have a positive culture from at least 1 bronchoalveolar lavage, or you need to have a lung biopsy with, well, typical lesions of Mycobacteria and a positive NTM culture. If you find that you are dealing with a clinically relevant NTM infection and such a patient needs treatment. An example, Mycobacterium szulgal. In the period of 1999 to 2006, there were 21 patients in the Netherlands of which we isolated Mycobacterium szulgal. 86% were male, and the mean age was 56. 71% of the isolates were from pulmonary material, from sputum, blood, etc. 67% were known with pulmonary disease. They were suffering from COPD or had a history of TB. 38% had systematic impaired immunity. Some had HIV, some had an interferon gamma receptor deficiency, and some were treated by anti-TNF or with steroids. And well, of the patients of whom we had M2G isolates, 73% met actually the ATS criteria. So we can conclude that at least in our country, isolation of Mycobacterium szulgal is usually clinically relevant. That does not mean that in other areas it's exactly the same picture. You have to do this type of research everywhere, because there may be other types of patients and other types of bacteria prevalent. Well, regarding patients infected with Mycobacterium szulgal, the symptoms varied significantly. Some were having cough, mucus production, tiredness, and weight loss. With regard to the X-ray and CT scan, we saw cavitation and pleural thickening and scars of pre-existing pulmonary disease. 71 had pulmonary isolates as stated, but we also saw some skin isolates. Mycobacterium szulgal can also cause skin lesions, but also from other materials like joints, pleural fluid, and even from feces. Interestingly, 19% of the patients had a positive microscopy, a positive Ziehl-Neelsen staining. Do you know the susceptibility, the sensitivity of Ziehl-Neelsen staining? Does anyone know? You need at least 10,000 bacteria per ml before you even can see them in the microscope. So they had really a huge burden of M2G. Well, regarding treatment, there's also a lot to be said about that. 11 patients that met the ATS criteria rightly received treatment as shown over here. However, 5 other patients that met the ATS criteria did not receive any treatment. 1 patient that did not meet the ATS criteria did receive treatment, and 4 that did not meet the criteria were rightly not treated. For patients who were treated, they received 12 months of rifampicin, ethambutol, and clarithromycin, which is a good choice. 2 patients died during treatment, but we are not sure whether that's related to the szulgal infection. And we saw a cure rate, if you may speak of that, regarding such a small number of patients. We saw a cure rate of 81%. Well, we did actually the same for all NTM species shown over here. And then we saw a very interesting picture. You see that the contribution of males to the whole cohort is higher for all species except for Mycobacterium simiae. For unknown reasons, well, we see less MCMA infections in males than of other NTMs as shown over here. The age was around 60. However, there were 2 species, Mycobacterium malmoense and Mycobacterium processus, for which the mean age was lower as shown over here. And an important conclusion, the most important conclusion, was that the percentage of patients meeting the ATS criteria was significantly different by species. There was 1 species, Mycobacterium novio-magense, I will come back to this species later on, of which none of the patients met the ATS criteria. And there were some species that were highly relevant, like Mycobacterium malmoense. In that case, 75% of the patients met the ATS criteria. And for instance, for Mycobacterium gordonei, which is also frequently found in tap water, but only a low percentage met the ATS criteria. Here you see it in a visual way. If you isolate bacteria of the tuberculosis complex, well, in 100% of the cases you have to treat the patient. If you isolate Mycobacterium malmoense or Mycobacterium szulgal, in our country, in 75% of the cases you have to treat the patient. For M. xenopi and M. kansasai, it's in about 50% of the cases, well, etc. And if you find Mycobacterium novio-magense or Mycobacterium gordonei, well, they are hardly ever clinically significant. So this is just a guide for the clinicians to be aware of a possible clinical relevance or, well, to neglect that. Some words on treatment of NTM disease. Well, there's a clear distinction in treating an infection by a slow grower or a rapid grower. Slow growers should be treated with rifampicin and ethambutol for 18 to 24 months. And the addition of clarithromycin and fluoroquinolones is still controversial. Clarithromycin is, at least in vitro, the most active drug. But with regard to the first-line anti-tuberculosis drug, there are discrepant results regarding the in vitro and in vivo activity. So there's still a lot to be examined with regard to that. For the rapid growers, you should use clarithromycin and parenteral drugs, aminoglycosides and carbapenems, for 4 to 6 months. For infections with rapid growers, we do rely on in vitro drug susceptibility, although evidence is also lacking at the moment. And you should not use the first-line anti-tuberculosis drugs to treat infections by rapid growers, because they simply don't work. We still sometimes see that in our country. I'm a little bit annoyed by that. So, in general, you can say that treatment of NTM infections are poor, worse than for tuberculosis. Infections by Mycobacterium processus are most difficult to treat. Sometimes you simply have to treat the underlying disease, like COPD, HIV, or leukemia. We have some publications on the use of surgery, as that is in some cases beneficial, but not all. And if it's not possible to cure the patient by anti-tuberculosis drugs, well, you should aim for suppressive treatment to have a particular quality of life. But we really need new drugs, new pharmacokinetic studies and clinical trials because, well, actually treatment is not very well arranged regarding NTM infections. At the moment in the Netherlands, for instance, we have pharmacokinetic studies in patients to see the variation in blood levels and that kind of things, and to compare that to the MICs in vitro, etc. Well, seeing the contribution of NTM infections to disease mimicking tuberculosis in the Western world, how is the situation in Africa? Well, we did studies, for instance, in Zambia, but also in Nigeria, in Kenya, and Ethiopia. For instance, in Zambia, we examined a cohort of 300 so-called TB patients. These patients, of course, were diagnosed by ZN, so microscopy and X-ray. We added our sophisticated mixed culture system and a molecular identification to see whether culture would support the diagnosis of TB. Well, in 50% of the cases, tuberculosis was indeed isolated, so culture confirmed the diagnosis of TB. In 29%, however, of the so-called TB patients, we could not isolate any Mycobacterium. In 21% of the TB patients, well, we could only isolate NTM. NTM isolation was associated with HIV positive status, and therefore not a result of cross-contamination of pseudo-infection and that kind of things. Well, these findings have been confirmed now in other African countries, and I think especially in HIV positives, NTM confused the diagnosis of TB significantly. And we therefore think that the diagnosis of TB should be refined. At the moment, WHO is organizing a lot of demonstration projects, for instance, with the gene expert, that in 2 hours' time can identify Mycobacterium tuberculosis in sputum and simultaneously detect rifampicin resistance. And I think that is very promising, although there are quite some difficulties associated with the introduction of such a sophisticated system in high prevalence settings. And I think NTM treatment needs to be introduced in Africa, which is a huge challenge, of course. So, risk factors. Well, I was mentioning HIV positive status, and it is indeed a significant factor. But there's another factor to be mentioned. We compared groupings of patients that used surface water from a lake, and the other grouping using recently introduced tap water systems. And the patients that were using tap water systems were at a significantly higher risk of contracting an NTM infection. So I really think that tap water is a risk factor for contracting an NTM infection. Some words on the identification of Mycobacteria. Until 1999, we still used biochemical identification. People had to do a lot of biochemical tests and also compare the growth characteristics of the bacteria, and within 4 or 5 weeks they were able to produce a result, which was of course a terrible situation. Since 1999, 16S DNA sequencing was introduced, and in that approach you actually sequence 150 base pairs of the 16S genes. There is quite some variation in that gene, and you can group the Mycobacteria according to that sequence. I remember visiting Professor Erik Duttger, who was still working in Germany at that time, and I asked him, well, can we really rely on identification on the basis of 16S sequencing? And he answered, well, you don't need to kill somebody twice. So he said, well, it is really sufficient to do that. Looking back at that answer, I think it's not completely true, and I will try to convince you of that. Since 2004, we use the reversed line blot identification to identify Mycobacteria. So we just amplify a part of 16S gene or a part in between the 16S and 23S gene, and we denature, of course, the PCR product and bring it on the strips with probes specific for particular NTM species. And well, within a few hours you can identify the NTM species that are most frequently encountered. So the system in itself works. But now a few examples why I think it is not sufficient. For instance, in 2005, 2006, we had a lot of commotion in the Netherlands about so-called bird tuberculosis. There were a lot of children suffering from lymphadenitis. And well, we took some lymph node biopsies, and from 138 out of 210 children, we were able to isolate Mycobacteria. And 94 out of 138, or 70%, were suffering actually from Mycobacterium avium. In the Netherlands, the second cause of lymphadenitis is Mycobacterium haemophilum. That's also frequently seen, but very difficult to culture. People usually identify them only by PCR. Well, because especially the newspapers were suggesting this was bird tuberculosis, we performed IS 1245 RFLP typing. Now in IS 1245 RFLP typing we can distinguish between different groupings of M. avium. The M. aviums that we find in humans and in pigs have a high degree of similarity, as you can see over here. If you sort them into clades or genotype families, well, they all resemble the same type. All 49 isolates that we have tested from 29 different bird species, they all have this characteristic pattern consisting of 3 bands. So this is typically the bird type pattern. Falkenham from the United States also isolated Mycobacterium avium from tobacco and filter paper. And well, we found this type of pattern of that type of an avium. So this does not seem to have anything to do with the M. avium bacteria that we isolate from humans. So with regard to this, you can just keep on smoking, no problem. Well, because of that clear distinction, we some time ago introduced a new subspecies actually. We proposed to reserve the designation Mycobacterium avium to the bird type isolates, and Mycobacterium hominis suis to the isolates from human and porcine sources. Well, we performed IS 1245 RFLP typing of that lymph node and avium isolates, and all were of the M. hominis suis type. So there were no bird type isolates among these isolates. So there is no bird type tuberculosis. It's simply not derived from birds. So this shows that sometimes you need an additional molecular typing to distinguish between different types of infection. On basis of the simple identification, it's just M. avium, but on basis of additional typing, it's 2 completely different subgroupings. Actually, if you have a look at identification of NTMs on basis of the 16S gene, well, all isolates from a particular species are identical, or you see very minor differences. So, for instance, here in the M. gordonii group, but generally they are, well, almost identical. However, if you also analyze other housekeeping genes, like the ITS sequence, HSP65, RQB, etc., you suddenly see a lot of variation. If you look at the dendrogram based on polymorphism in these genes, well, you see that they start to fall apart. And that again shows that there's a high degree of divergence within species. With our simple molecular tools, we group isolates within NTM species that are highly diverse. and they have another clinical relevance. So we need an improved identification. An example to prove my case. We asked ourselves whether the grouping Mycobacterium xenopi is valid to serve the clinicians. As stated, in 1999, the RIVM started with 16S sequencing for NTM identification, and we actually had two xenopi subtypes, genetically highly related xenopi type 1 and 2, but also another one that we designated unknown Mycobacterium species number 1. Our report to the clinicians at that time point was xenopi-like. So we made the suggestion that although this bacterium was a little bit different, it actually resembled the grouping of xenopi. First, some words on xenopi 1 and 2. We had 45 patients with pulmonary isolates. 76% were males with a mean age of 60 years, and 63% were suffering from COPD. We had 2 patients with pleuritis and 2 patients with spondylolisthesis. 21 out of 45 or 47% met the ATS criteria. The patients of whom we isolated xenopi type 2 were more frequently clinically relevant than the other ones. So the distinction between type 1 and 2 in itself is already important. And is that unidentified Mycobacterium species 1, that xenopi-like bacterium, really an xenopi? Well, we first compared the colony morphology. xenopi 1 and 2 had very small yellow colonies as shown over here. For the xenopi-like bacteria, we saw wider and larger colonies as shown over here. Actually, we observed, and we did already know, that one base pair difference for all the isolates. So is this distinction actually clinically relevant? Well, to answer that question, we had a look at the 17 patients of which we isolated xenopi-like bacteria, the UMS1. These 17 patients were for 77% males. The average age was 53 years. 88% had a pre-existing lung disease. They were all living near the place of Nijmegen in the western part of the Netherlands. And of this grouping, none met the ATS criteria. So that is why the description of such a species is important. In case you isolate xenopi, well, in nearly 50% of the cases it's clinically relevant. If you isolate Mycobacterium novio-marchense, well, it's almost never clinically relevant. Actually, we described this species on the basis of biochemical tests, cell wall analysis, etc., molecular characterization. Our conclusion was that it's closely related, but actually a separate species. And so nowadays it's possible to distinguish this grouping from xenopi, and that serves the clinic. So what is the future of identification? What's actually the future of the whole laboratory diagnosis? Well, I think we should use whole genome sequencing. We at the RIVM already have analyzed sequence data from 200 tuberculosis strains. Problem of sequencing is non-existing anymore. Sequencing goes very fast. That is not a problem. Data analysis is very cumbersome and takes weeks or even months. However, as soon as we have developed our algorithms to extract the relevant information out of that huge amount of information, I think it's possible to identify the bacteria, to predict the resistance, and also to have an extended DNA typing actually of isolates. So, yeah, I think this is really worth exploring. And that is why we propose to have a project in the European Union, actually whole genome sequencing of 1500 NTM isolates. This is something in which Professor Drancourt was involved. He was very enthusiastic about this idea of sequencing 1500 NTM isolates from the environment, from animals and humans, from different parts of Europe. It should serve an improved identification of NTM. Actually, what we want to do is to build a new, robust phylogenetic tree of all NTMs. So to have an improved taxonomy, to be able to associate clinical relevance to minor branches of the tree. And in that case, if you isolate such a species again, you can already predict whether it's clinically relevant or not. Unfortunately, that project application was rejected at this time point. It was not really fitting the goal, but it's ready for resubmission. We will surely resubmit it. So the conclusions. Well, NTMs are frequently found in humans, and the isolation in many parts is increasing. The clinical importance is often unclear and generally also underestimated. We have shown that the relevance differs significantly by species according to the ATS criteria. Because we introduce liquid culture in many settings, and also in Afrika, we need to have a better identification of the bacteria. Otherwise, the tuberculosis diagnosis is also heavily influenced. We need more taxonomic research to have a better taxonomy of the NTMs. The treatment of NTM infections is at the moment partially not evidence-based, and that is really a problem, because treatment outcome is poor, and there is a need for clearer guidelines regarding the diagnosis of NTM infections and treatment. And we are putting that together in the Netherlands at the moment. On European level, people have also established a new network, the NTM-net. It was actually an initiative of our neighbors, the Germans of the University of Freiburg. And well, they are promoting the type of research I have shown to you in this presentation. So we try to have more insight into the clinical relevance of NTMs. Well, this is our grouping. Thank you very much for your attention.
Questions?
I have two questions. So, do you know the source of the contamination from the children with foods?
Well, we presume that they are infected by contact with soil. They just play in the sand and that kind of things. We easily isolate the same type of Mycobacteria that we find in the lymph nodes from the soil. So we assume as soon as they get into contact with soil, well, that they are infected. But also regarding pot plants, the soil in pots is also heavily contaminated with Mycobacterium. We have done some isolation of that in several homes. So if children are just curious and they start to plucking plants and that kind of thing, actually Mycobacterium likes cellulose. So Mycobacterium is also present on leaves of plants and that kind of things.
And my second question is, do you think about phage therapy?
Well, phage typing is interesting because with phage typing you can also distinguish between many different types, and it also gives you an impression of how many groupings are present in a species. However, seeing the possibilities of whole genome sequencing, I think we will have many more different groupings than we could have with phage typing. Question is, how many groupings do you want within one species? But if we have that new phylogenetic tree, we can just add all new sequences to it, and it will become more and more reliable. It will grow. Yes, please?
You talked
What types of...
What was the threshold or what did you find when you studied for East Africa, in particular Kenya? Because I just saw you were talking more about Zambia.
Yeah.
I didn't see the results for Eastern Africa.
No.
I'm just interested.
We partially did find isolates of the same species as we recognize, but that's only based so far on the rough identification, so the identificational basis of just a small part of the genome. And also for that reason we need to introduce whole genome sequencing to know whether isolates from that part of the world are really the same as isolates from our region. On basis of the rough identification, we also found an avium and a lot of other mycobacteria that we are familiar with. But I doubt whether these bacteria are truly the same bacteria as we find over here, because we do see a lot of difference in clinical importance of isolation in different areas. For instance, Mycobacterium avium is clinically relevant in our area, but not so much in the United States. And intracellular is highly important in the United States, but not in our area. So I think we are not referring to particularly the same type of bacteria. Our identification also in that case needs to be refined.
Could you identify specific virulence factors which explain the differences in pathogenesis of these species?
It's of course a very important question. There is a predisposition, a difference in susceptibility to NTM infection. And we know only very, very little bit about that. There are differences, abnormalities, in particular receptors of the immune system. But we have only found a very small number of these differences. And I think we should look much harder to explain the differences in susceptibility. By the way, I was explaining that previously people thought that tuberculosis was an inherited disease. To some extent, it is an inherited disease. We see that in particular families you have much more breakdown to disease after infection than in other families. So that there is a predisposition, but we don't know much about it now. If you look in the literature, it's very, very modest what people know about that. Just a few incidental things have been observed, but we don't know why someone is more susceptible than another one, except for having an underlying disease. Of course, that makes you more susceptible. But regarding the functioning of the immune system, there's a whole field to be investigated.
Do you start to use multi-topics to identify?
Yes. That is an interesting point. So far we use, single target identification based on 16S or the target between 16S and 23S. Some people use the RPOB. Nowadays people switch to the multi-top. So that has to do with a whole profile actually of the bacteria. And the big question now is, does that give a more reliable identification than the single target identification? People are having a lot of research into that at the moment. Yes. It's at least fast and it's cheap. And I think it's not less discriminative than the identification that we use nowadays. But it gives you some differences in identification. We don't know exactly how to deal with that.
Thank you very much.